All right, we're going to cover the various types of T cells. We're going to begin with cytotoxic T cells. These are, you'll see them referred to as like a capital T with a subscript C, or killer T cells. But I just call them cytotoxic T cells. That's what I'm used to. So they are CD8 cells. And they're responsible for cell-mediated immunity. The activated cytotoxic T cells divide into more, and then also they will form memory T cells once they're activated. And what they do, they destroy bacteria, fungi, and transplanted tissue by releasing perforin that ruptures the target cell's membrane. So that in that way, they're similar to the NK cells. Then they also secrete cytokines that activate apoptosis in the target cells. And they secrete lymphotoxins, which disrupt the target cell's metabolism. So indirectly will kill the cell or inactivate it. So cytotoxic T cells basically kill cells that are possible, different types of pathogens, So the steps in the activation of cytotoxic T cells, first is antigen recognition. This is your infected cell here, and this is your inactive cytotoxic T cell. You can tell it because it's a lymphocyte. It has that big nucleus that almost fills up the whole cell. So you have your inactive cytotoxic T cell, has a receptor that is able to bind to the antigen on either a viral or bacterial antigen in an infected cell. So, and since cytotoxic T cells are CD8, they're going to bind to class MH1 proteins. So that would be an antigen presented on any cell with a nucleus. And then we have step 2, which is activation and cell division. The antigen recognition and co-stimulation by the CD8 and class 1, interaction. That is going to cause those cells to divide and produce more cytotoxic T cells and also memory T cells. And then step 3 is destruction of the target cells. So that's where the cytotoxic T cell is actually going to completely destroy the target cell in those various ways that we discussed. Now, a helper T cell is a CD4 T cell, it has a CD4 marker. So it's only going to be activated by class 2 MHC proteins. Remember 18 and 24. The 24 means that class 2 and CD4, okay? So that means your CD4 T cells are only going to bind to antigens bound to class 2 MHC proteins. And that means that these are going to be the antigen presenting cells, either dendritic or macrophages that have presented an antigen on their surface. So helper T cells are also called TH cells. They are CD4 T cells. They are activated by exposure to antigens bound to class 2 MHC proteins, meaning APCs, dendritic or macrophages mostly. Now, once they're activated, helper T cells divide into memory and active helper T cells. The active helper T cells secrete various cytokines, like interferon is a cytokine, but various cytokines. And then helper T cells, and this is where it really comes in how important and how necessary that they are to immunity. Helper T cells stimulate both cell-mediated and antibody-mediated immunity. So helper T cells have to be activated before cytotoxic T cells and also before B cells will become activated plasma cells. So memory T cells, There are both cytotoxic and helper T cells that can become memory T cells. And that, of course, that's going to be important in any subsequent exposure to that same antigen. Regulatory T cells are CD8, just like the cytotoxic, and they will suppress the response of other T cells and B cells. They secrete cytokines called suppression factors. Remember, we already talked about this, that it's important to regulate the immune response as well. We don't want it going overboard. And checkpoint question 17, where are class 1 MHC proteins and class 2 MHC proteins found? Class 1, any cell with a nucleus, and class 2 will be antigen-presenting cells, such as dendritic cells and macrophages. Now, B activation and something called sensitization, which I'll explain to you. So each B cell type has specific antibody molecule in its plasma membrane. So each B cell type is going to have one of the 5 different immunoglobulins, IgA, IgD, IgM, and we will go over those. When the matching antigen appears, the antibodies bind to the antigens. Antigens enter the B cell and are displayed on the surface, bound to class 2 proteins, because that B cell is now an APC. The process is called sensitization. And then the B cell is activated by helper T cell via cytokines. So for a B cell to be activated, and it has to be sensitized also, for that to happen, the antibodies that have already bound to antigens, those antigens have to enter the B cell, and then they're displayed bound to class 2 proteins, and that makes the B cell now an APC. And then after that, before the B cell is actually activated, it also must be interact with a helper T cell. Now, activated B cells will divide into 2 things: plasma cells that secrete antibodies and memory B cells. And this is the figure 14-14 shows you sensitization. Here's your inactive B cell that is coming up to an antibody that has an antigen attached. Okay. I'm sorry. Start back over here. Here's your inactive B cell. Here's the antigen that's binding to an antibody. And then it's going to be expressed on a class 2 MHC protein. All right. Once that happens, it's called sensitized. So this picture here, where it says sensitized B cell, notice that here, this is the Y-shaped structure is an antibody, and it's got the antigen attached to it. Another copy of that antigen is now being expressed, and this is your MHC class 2 protein. So now we can call this an APC, an antigen presenting cell. The next thing is the sensitized B cell has to be activated by its interaction with a helper T cell. So you can see where it's blown up here, the class 2 protein on the B cell with the antigen attached also binds with the T cell receptor, which is a, it's not on here, so ask yourself, is it CD4 or CD8? All right, this is class 2. Remember the number 24. So class 2 would always interact with a CD4 T cell receptor, so marker. So anyway, after that part, then your activated B cell will divide into plasma cells, which produce antibody, and memory B cells. Now, as far as the antibodies, how do they actually work? Because B cells destroy antigens or attack antigens by producing antibody, but it's the antibody that does, the job. So antibodies have constant and variable segments. And it is the variable segment that makes it one of the different types, Antigen binding sites are free tips of the variable segments. and they determine the specificity of the antibody. So there's the antigen binding site is specific to the antigen that it's going to bind to. This is an illustration of a typical antibody structure. Notice the Y shape and the antigen binding site here and here. There are two antigen binding sites on the ends, the top ends of the Y. And they're here your variable. This is the location of your variable segment. Okay, so if this brown glob is your antigen, it's got these little pieces of it, portions of the antigen, these little yellow pieces that are called antigenic determinants. And those are the portions that actually the antibodies will bind to. The antigen binding site will bind to the antigenic determinant, which is the piece of the antigen that the antibody binds to. So that's how it looks when an antibody binds to an antigen, forms an antigen-antibody complex. And again, in the notes, it makes it clear antibodies bind to portions of antigen called antigenic determinant sites, also called epitopes. And here are the classes of antibodies. There's 5 classes, and they begin with Ig because antibodies are also called immunoglobulins. All right, so make sure you know the 5 classes. IgG is the largest class. It can cross the placenta for passive immunity for the fetus. IgM is the first antibody produced. It is responsible for cross reactions or transfusion reactions in blood types. IgA is found in secretions like tears and saliva, exocrine secretions. There is a question that asks you if it's you have to know it's not endocrine secretions. It's exocrine secretions like tears and saliva. IgE is involved in the inflammatory response and allergies. You also would see it. IgE also stimulates basophils and mast cells to release histamine, and that, of course, is going to trigger the inflammatory response. And IgD attaches to B cells and aids in sensitization of B cells. This is the table that goes over all of the classes, and this is slide number 74. You may want to find it in the PowerPoint slides and just read through the classes and then their remarks about the classes, because it goes into a little more detail. Now, 6 functions of antibodies. So this is how they actually work in our immune system. First is neutralization. Once the antigen and antibody forms the antigen-antibody complex, that prevents the antigen from attaching to a cell. So it neutralizes it. The second thing is precipitation and agglutination. So precipitation is when the antigen-antibody complex settles out of solution. And agglutination is like what we saw with the wrong blood type being donated to a recipient. It caused clumping of the red blood cells. Okay, that's called agglutination, and that can happen when antibodies are coated with, antigens are coated with antibodies. Then the third function of an antibody is to activate complement, and complement is part of the innate immune system. And remember how we talked about that the innate and the adaptive immune systems are independent of each other, but they also work together. So here's an example where you have the adaptive immune system causing activation of part of the innate immune system. So what happens when complement is activated is it binds with, and so once the antibody binds with the antigen, it changes the antibody shape, and then it can bind with complement. And that just helps with different ways. Complement, remember, complements or adds to the activities of the immune system. Let's see. So Binding of an antibody to an antigen will attract phagocytes to the area and enhance phagocytosis in this process called opsonization. And then as well, antibodies will stimulate basophils and mast cells to release histamine, and that stimulates inflammation. Now, this table 14-2, which is slide number 77, lists the cells that participate in tissue defenses. So it goes over neutrophils, eosinophils, which are both phagocytes, mast cells, and basophils, which both release histamine and heparin. And then antigen-presenting cells are macrophages and dendritic cells. They are the ones that contain the class II proteins. And then you have all of the lymphocytes, NK cells, which are part of the innate defense, and then all of your T cells and B cells are listed and their functions. So this is slides 77 and 78. Both are pretty important.